Swayam Prabha. Digital India. Educated India.
Then these PDB structures, they have one more term. So, I will show you here. So, if we have the XYZ coordinates, this occupancy, right, then we have another one term here, right. This is called a temperature factor. Right. Also we can call this as the B factor. This will tell you the flexibility of each atom. How to get this one? So, if you are able to fix the atom within the electron density map, if you fix the atom within that space, right, they are rigid, and in this case they do not disturb these electrons. So, in this they have an ideal situation. In this case you can get the dense distribution, right, within that particular conformation, then they are very rigid. But usually the electrons have wide distribution, right, because right, then they vibrate. Because the vibration of these atoms, they have the different molecules in the crystal lattice. In this case, you can see the electron density, they will include all the average of the motions. If they vibrate more, then you can see the deviation is very high. If you can occupy the same residue or same atom within that rigid position, then this is very small. If you not, then they fluctuate very much. In this case, you can see the variation of these fluctuations there, that is what they give in terms of temperature factor. Okay, we see this case, right, you can see the histidine 93, okay, you can see this contour can accommodate this particular residue, and some case the contour is not able to accommodate the full residue because it is vibrating more inside this map. So, you can get this information, right, and account these motions in the PDB structures, right. This is what they give you the data B factor or the temperature factor. Essentially, if the number is less than 10 or less than 20, then we can say that the vibration is less and they are rigid, right, and then they have the particular position in the structure. If it is more than 50, then the atom is moving here and there and the fluctuations are more. In this case, the temperature factor or the B factor is high in this case. So, this is one example. So, here you can see the histidine 93, ND1 and the CD2. The value is around 12 or 15 or 16, and you can see these atoms, they are rigid. And the same case, if you see the histidine 81, right, ND1 and CD2, right, here you can see the values are more than 50, say 72 or 73, and in this case you can see that these atoms, they are highly flexible and these are the B factors are very high. So, here I show the picture. In this case you can see different colors, for example, which is the blue color, or some of them are in yellow and red. So, the blue color ones, right, they are having low values, right. Low values means what is the meaning of low values?
Rigid, low flexibility.
They are rigid, not low flexibility, they are rigid. So, some cases if it is yellow or it is in red, in this case they have lot of motions, right, and in this case they are highly flexible. So, if you see this data, right, for example, I show the structure right here. So, which is they are highly flexible because the values are very high in this case, because this is from the DNA and this is from the protein side. And some cases if you see here, and many cases what is this, right. So, here there are less, then this case these atoms are rigid. So, now we get the 3D coordinates, right. Now, I will explain about the various options available in the protein data bank. So, first we have the sequence. When you get the sequence information, right, you can see the amino acid sequence here, right. Earlier I showed the FASTA format, you can see the PDB here, as well as you can see the secondary structure assignment. What is the meaning of this spiral ones?
Helix.
This is helix and this is strand, right. This is based on the DSSP. What is DSSP? Dictionary of secondary structures of protein. So, they take the PDB structures and based on the hydrogen bonding pattern, it will assign the secondary structures, right. So, here given the amino acid sequence, you can see the helix, strands and so on. So, different secondary structures. So, now go to these different annotations. The proteins are classified in different groups. I will explain the details maybe in the next class, right. In some cases which are dominated by helical regions, In this case you can see the proteins as helix proteins, and some cases you can see the protein is dominated by beta strands. These proteins we call as beta proteins, and some proteins we have both alpha helices and beta strands, they are called alpha beta proteins or alpha plus beta proteins or mixture proteins. So, there are some databases which have this information, right. If you see the annotations, you will get the information regarding that, right. I will explain more detail in next classes. So, you can see the sequence similarity, right. The PDB recently revised the page. So, for each similarity, for example 100% or 90% or 50%, they developed several clusters, right. We discussed about the clustering method, right. Which clustering method we discussed? Right, sitting in which clustering method? K-means clustering, right. So, use the different clusters, they keep all the sequences together and they put the similar sequence in one cluster. So, they can make different clusters depending upon any thresholds, right. So, they have clustered various clusters, right, with respect to the identity, and you can see from these clusters that they get the non-redundant sequences. They have the sequence similarity you can get for each proteins, right, in the PDB. Then you go with the 3D similarity. What is 3D similarity? The similar in the structures. So, now we discussed about the sequence similarity. In this case we have align the sequence 1 with the sequence 2 and see where they are similar. In the case of 3D similarity, they take one protein structure and they superimpose this protein with the other protein structures, right. We will discuss the details later and see how far they are different depending upon a C alpha position or all atoms, right. They define based on the term RMSD, that is root mean square deviation. So, here we have the coordinates x1 y1 z1 and x2 y2 z2. They can get the distance when they assign to superimpose the structures. There are several atoms, right, or several C alpha atoms. For each case we calculate the distance and finally they get the root mean square deviation, right. This will tell you how far these two structures are similar. If the structure is similar, then the RMSD is less or high? Low, right. If it is less, you can see the structures are similar. If it is very high, then we say that they are not similar, fine. So, you can have the values, right. So, these are the structures, right, which can have the similar structures, right. So, in this case they align with another chain on XX7 and to give this root mean square deviation, right. This is 0.47 or 1.29, right, they are very close to each other, right. Then we go to the literature. Literature will give you the information on the papers which have been published. So, based on different techniques or different stability or different information, right, in the literature. For example, for this structure T for lysozyme, right, there are various papers published on this aspect. So, they listed up the primary citations for that particular paper. So, then if you see, they also linked with the other papers which used the PDB structures. For example, there are several other methods, right. So, for example, this fold rate and getting critical residues and the effect of proline mutations on stability, right. So, there are various methods. These papers they use the PDB structures for developing a model, right. So, there are several papers they use the structural information, right, for the analysis. They listed up all the papers in the site. So, if you click on this one, right, so then if you see these are the structures which are used in the paper. They also give the data about the structures they used in a particular paper, right. So, this will give you the PDB is important, PDB structures are important and the PDB structures are widely used for the analysis as well as for developing prediction methods. So, you give the biology and chemistry. So, you can give the structural keywords, right, as well as the description of the proteins, the weight and the source method and so on, right, and you can get the information regarding the GO terms like the cellular location, molecular function or biological process, and you can feed these enzymes and you can get the catalytic site residues, right. So, what is catalytic site residues?
The residues which perform catalysis.
Catalysis, right, this shorter small segments, right. So, you can see for example, which is the database you get the catalytic site residues? Catalytic site atlas, right, you can see CSA, right, you will get this information. You can see these are the residues, right, which are important and acting as the catalytic site residues. So, then X-ray diffraction they will give the crystal data. For example, if you have any crystals, they have the length ABC and the space groups, right, and they give all this information as well as the number of heteroatoms, solvent atoms refinement, and then here you can give the bond length, right. There are various types of atoms like between C and N and C and O, C and C beta, right. So, different types of this bond length they give the average and the standard deviation. For example, if you have C and N, what is the bond length for the C and N? So, that is 1.33, right, because C and N, right. So, you can see 1.33 and the standard deviation we give, this is a minimum 1.27, this is 1.38, right, this is the minimum and this is the maximum, fine. So, in case of bond angle, how many atoms are required to get the bond angle? 3, right. So, you can put C and C alpha, right, if it is C and N and C alpha, right. So, you can see this is the average, there are 149 angles, 129.87 is the average. So, the deviation is also not much, right, 114 to 133. So, this is a minimum value, this is a maximum value and you can see the average as well as the deviation. Then you get torsion angle. So, we discussed earlier about torsion angle. So, you can see the 4 different atoms, right. So, the 5 angles, right, you can hear the average values, the minimum and the maximum values are here and you can see the average value for the different dihedral angles. Then we go with the methods and the geometry, right, as well as they give the links with the structural summary, structural features, ligand features and so on. So, you can get all the information, right. So, when you go to the PDB, right, rcsb.org, right, then you can get all the information regarding a protein. So, just you try with any of the PDB ID, right, and you can read this, see the details which are available in protein data bank, right. So, there are classifications and then biological details and other information regarding the pathways and motions and so on. So, the PDB has several options to search to get the data. First, you can use the simple search. In this case, you can search with the any PDB ID or you can the names or any keywords. So, you can use any of this information in the simple search and you will get the information. And they have the advanced search. In this case, you can search with various other options. You can check with the ID and keywords or the structure or any type of annotation or the molecular type, sequence features, chemical compounds and methods. For example, if you want to get the data for all protein DNA complexes, right, if you are interested in the protein DNA complexes, right. So, in this case, which keywords you have to search, which option you have to search for to get the protein DNA complexes? This is a molecular type, because there are several molecular types and I showed at the beginning there are many proteins and several nucleic acids as well as some protein nucleic acid complexes. If you want to have the protein complex, you can select the proteins and see whether you need the monomer or the dimer or the trimer and so on, right. If you are interested in protein DNA complexes, right, you go to the molecular type, now you can get the, okay, next box will open. So, we have the questions. So, first the question is whether it contains protein, right, yes or no, yes, because we need protein DNA complexes. So, this is yes and it contains DNA, you need yes, right. So, you put yes. Then it contains RNA, no, right, because here we do not need this RNA because we are interested in protein DNA complexes. Then whether it need to contain protein DNA and RNA hybrid, there also we do not need because we are mainly interested in protein DNA complexes. So, if you want to have the protein DNA complexes, you search for the proteins and DNA. So, if you do this, right, you can get the data. In this case, you have also another option, whether do you need to remove the sequences with some redundancy. If you want to remove, you can also remove the redundancy, you can set some cutoff identity. If you do not want, just you do not have to remove. If you want to remove, you have to put this and then there are several options, you can click which redundancy do you want. Here I put 40% identity, so now we get the data. So, here also they classify different groups, you can see the organisms, you will get the Homo sapiens 538 and equal to 48. So, you can make the classifications and you can analyze separately if you are interested. Then you have the taxonomy, different methods and resolution. If you want high resolution structures, you take these, right, you will get the good number of structures. Then you are interested in latest data. So, you can have the data which are very latest. So, in this case you get this month they got 2 structures deposited in database for the protein nucleic acid DNA complex, right. Polymer type is protein DNA. Then we have different enzyme classification. So, these are the different classifications and different types of proteins, right. This I will discuss later about the scope classification. So, if you want to have a group of proteins, you can get the group with different aspects, you can with the different organisms, different taxonomy, different methods, different enzyme classification and so on. If you want to have data on protein complexes, then what to do?
On protein.
You can get the proteins, but you need to have the complex, means at least we require 2. So, in this case you can select the number of chains, right, minimum 2. If you want to have the different proteins, not the dimer or homopolymer, if you want the heterodimers or heteropolymers, then if you have to mention the chain length should be different, then you can get the heterodimers or heteropolymers, right. So, likewise you can get the information and collect the data from the protein data bank. So, now if you have these complexes and you can get lot of information from the proteins, right, and you can also use these structures to obtain various parameters, right, and you can also visualize the protein, right. The protein structures I shown a static structure, right, this is a kind of static structure. So, you can see only one motion, right, this is only one view. So, you can view this protein and you can view in different directions, and also you can observe various information from these particular proteins, right. So, there are several software available to visualize the proteins and to get the information from these protein structures. The commonly available software for visualizing proteins, right, the PyMol and RasMol, JMol, okay, King and WebMol, they are developed earlier days, and Swiss PDB viewer. So, RasMol is one of the oldest ones, right, just to view these protein structures and to understand how they make the contacts and what are the interactions and all these things, right. Currently JMol, Swiss PDB viewer and PyMol are widely used software to visualize protein structures, and among all these things PyMol is getting famous because can get the publication quality figures. And also there are several options available in PyMol, right. You can fruitfully utilize the PyMol for obtaining several information for a protein structure. I will show you a short demonstration, right, about the utilities available in the PyMol. Earlier days when they started the PyMol, it was free for everyone, right. Then after some time when it is getting famous, because many users started to use this PyMol software, then the Schrodinger bought this PyMol and they incorporate inside the Schrodinger software. So, they became commercial, right. However, they are kind enough to give this for free for educational purposes for the most of the utilities. So, if you want to get this PyMol for the educational use, first we need to register and you have to get the software, right, by giving the necessary information, right, to the developers. So, you give the necessary informations, then they will give you the key, right, and then you can register, right, and you can install the software, right, in your desktop. So, here this is the website where you can get the PyMol login, fine. So, once you get this PyMol, right, and then you start to use it. So, when you install and when you launch PyMol, you can get 2 windows, right. So, you can see this is one window, this is with the command prompt. You can use any command, right, in this space and execute the program. Then you can see the next one, the down. So, here this is the viewer window. So, here you can see your molecule and you can do some manipulations in this protein molecule, right. So, first we need a structure. So, it accepts the PDB and also several other formats. For the PDB, just if you go to the file and you take the option open, then it will ask you the file which you want to open. So, then you go to this your directory. So, if one tm.pdb, this is the one, if you choose, right, when you open it, right, then you can see this structure here, right, you can easily see the structures. So, default is lines. So, you can see several lines here. So, here it represents the PDB ID 1 TIM, right, this is the PDB ID which you can see in this picture, right. So, when you see the right side, then you can see these 5 letters A, S, H, L and C. A is for the action, this will tell you what you want to do, and S is for to show, right, whether you want to show the atoms or show the structures or not, and H is to hide. If you do not want to see, then you can hide, and L is to label and C is to color, right, and A is for the action, right. I will tell you a few examples to how we use this. So, first you want to show this as a cartoon. So, we use this S, right, you can see this is the S here. So, you want to show this as a cartoon, currently it is lines. If you want to show in cartoon, right, then you go to S and show as cartoon. If you do like this, then you can get the figure like this, it is a kind of cartoon. If you see this one, it is very difficult to see where are the helices and strands. Can we see this, where you can see the helix and strand? We cannot see that, right, it is very difficult. If you see here, then here you can see where the helices are located and where the strands are located. Now you can see the spirals here that represent the helix, and you can see the arrows that you can see these strands. Then also you can color by chains. If you see this one, how many chains in this structure? We do not know, right, maybe 1, maybe 2, right. If you color by chain, right, automatically it will color, right. Now you can see how many chains? 2, easily you can see, right. So you can use this information, color information, you can see now there are 2 chains, this is 1 and this is 2, right, in this chain. So now you can also hide, for example 2 chains here, if you want to hide one chain, so you right click on the chain, right, and you can get this information, for example chain information, and if you go to this hide, right, the chain and hide, so and click on everything, right, and you can see one chain is hide. Here 2 chains, so we select one chain, this chain we selected, and if you hide that chain, so now that chain is hide, then we can see one chain. It is also possible to zoom in and zoom out, and also you can change the orientations, also change the locations. You can move this mouse and you can change it to the different orientations, right, and you can have different views, as well as you can zoom in or the zoom out this particular structure. So okay, so now if you want to see the different secondary structures in different colors, for example if you want to see the helix, strand and the loop, so different color options. Here we use red as helix and yellow as the strand and the green as loop, right. In this case you can see this is the case, red is helix, right, you can see helix here, and you can see the strand in this yellow one and the loop, right, in this green one. You can clearly see the difference between all these secondary structures. So now if you want to display a sequence, now we are dealing with the structure. If you want to show the sequence and see where are the helices, in this case you go to the display option, right, you have the display option here, and here there is option called the sequence, right. If you show the sequence, then you can see the sequence. Also if you see this region corresponds to helix here, and this region corresponds to strand, and this region corresponds to loop, right. You can see the same color they apply to the sequence and the structure. So you can map and see where you are the helical segments, strands as well as the loops in the sequence as well as in the structure. So you can also change the color in terms of the chains, right. If you are to this is very long, if you drag through, then you can see one sequence in one color, right, and another sequence in another color. So now if you want to see a few segments, any segments, or if you want to measure any bond length or bond angle or torsion angle, so you have selected the segment 21 to 26, right. It is very simple, just to click on this sequence, then that is represented in the structure. You can see the same in the structure, the pink dots, these are the residues which represent the same in the sequence, for example L, G, E, L, I, H. So now you can see and you can zoom in to select the residues, you can see the pink ones, these are the selected residues, and you can hide all others. So if you see these specific residues in the ball and sticks, right, what is ball and stick model?
The bond.
Yeah, sticks represent the bonds and the balls represent the atoms. So if you see here there are different colors, right, they follow a specific color for any specific atoms. For example the green as carbon, and N for this nitrogen is blue, right, and the oxygen is in red, right. Okay, this is oxygen, right, this is the nitrogen, and you can see this is a carbon, right, you can see this. Now you can label the residues, right. So here this is here we have the residues, glue 23 is here, 23, leucine 24 and isoleucine 25. So this is the one we selected, so these residues are given here. So which is glutamine 23, it is marked here. So you can also now the selected leucine 24, and you can also mark with this atom name. Here we labeled this is C alpha, and we have C beta, C gamma, this is the main chain, right, and here this is side chain. Okay, it's marked here, these are the main chain atoms. What are the main chain atoms? NCaCO, right, so main chain atoms given this one, right, the backbone atoms, and here these are the side chain atoms, right, this is a leucine. So it has a side chain C beta, C gamma, C delta 1, C delta 2, right, this is shown here. Then you can also see in this line options and so on. It is also possible to measure the bond length, bond angle and torsion angle. So in this case go to YZ, right, and then go to the measurement, right, it will ask for the atoms to measure the distance angles, diagonals as well as the contacts. How many atoms are required to get the distance? 2, right. So, if you want to get the distance, it will ask the atoms, select the first atom, right, click the first atom. So, if you click on the first atom, right, say this one, right, and it will ask for the second atom. So, they click on second atom. So, now it is CD1 and the CG, right. So, what is the distance? It is 1.5 angstrom. So, you can see the display in this in angstrom, right. So, angstrom is equal to 1 10 to the power minus 10 meter, right, 10 to the power minus 8 centimeter, right. So, you can see this is the angle, this is distance 1.5 angstrom between CG and the CD1. Now, if you have this angle, how many atoms are required? 3, right. So, we have to select 3 atoms to get the angle. So, you can see that CD1, right, 1, 2, 3, right. If you CD1, CG and CD2, if you click 3 atoms, then this will give you the angle. So, the angle is 109.9 degrees. Likewise, we can use this pi mole for various aspects. You can see the mutations, you can see the molecular surface, and you can see the interactions, you can see the contacts, right. For example, hydrophobic interactions, electrostatic interactions. So, you can play around a lot with this pi mole. I will show one more example. For example, to see the B factors, right. We discussed B factor. What is the B factor? Is temperature factor. This will give you the fluctuations of each residues, right. For example, how much they are fluctuated and how far they are rigid. So, if you see this one, if I take this file, so I take one AQ1, right, they open this file in this lines, right. So, this is you can see on surface, right, this is the line 1 and you can see the surface, and from this you can see the B factors with the different colors. You can see the blue colors, this is rigid, right, that is less flexible, and you can see the orange ones here and there, right, here. So, you can they are highly flexible. So, we see many residues are rigid because they could accommodate the residues within the electron density map. So, in this case several residues are rigid and some cases they are fluctuating, these residues are highly flexible, right, they have more B factors. So, then if you have the pi mole, it has several applications. For example, you can see the comparing two structures whether they are similar or not. You can mutate any specific residue and see what is the changes because of these substitutions, and you can get the high resolution structures, and also you can get measure the bond angle, bond length and torsion angles. You can analyze various types of interactions, right. You can manipulate or you can play around this pi mole to get various informations. You can also get very high resolution figures, right. This is acceptable in publications, right. And another important fact is it supports Python script for the user, the necessity of these users, right. You can find the interface residues, pairwise distance and so on. For more information you can look into the tutorials, right. You can see the websites, right, and you can get more information, right, and you can use this pi mole, right, as an effective resource, right, in your project or in your reports or in your papers or anything or any applications. So, to summarize today's talk, so what did we mainly focused on? Protein 3D structures. 3D structures provide which information? Atomic information, explicit coordinates, right. So, what are the various experimental techniques used to determine 3D structures?
XRD and NMR.
X-ray crystallography, NMR spectroscopy. These are the widely used methods, right. Electron density is also now used for the determining structures, right. So, what is the resource? What is the database which contains protein 3D structures? PDB, right, protein data bank, right. So, this is organized by the different institutions, right, different places. Now, we have the worldwide PDB, right, fine. So, which information, what are the other information which can get the PDB? For example, if we take the coordinate file, the coordinate file gives the information on the atom number, atom name, residue number, residue name and the chain information and the coordinates, occupancy and the temperature factor. What is the occupancy?
How many in unit cell? How many?
That the orientations. The atom can take how many different orientations. If one orientation, this will be 1. Different orientations, the total sum will be 1. Then temperature factor, right. So, then we discussed about the visualization tools, right, and we gave a demo on one tool, which one we discussed? Pi mole. What is the application of the pi mole? You can get the structures, you can view the structures, you can see the various orientations, and you can map sequence and structures and the mutations and get different types of interactions, and you can measure the bond length, bond angle, right, and you get a lot of applications, right, for by using pi mole. So, we discussed more about 3D structures and the coordinates. Now, the question is we have a lot of data, right. PDB contains 133,000 structures, right. So, what can we do with these structures? What are the various information you can get? In the subsequent classes I will discuss about the parameters or the properties, right, which we can derive, right, from the known 3D structures, and how these parameters can be utilized for understanding the structure or understanding the function of these proteins as well as their complexes and so on. Thank you for your attention.